molecules, so the molecules that, or the genes that people type before transplants, for example, or self-incompatibility systems in fungi and plants. This is on the order of the polymorphisms you see there. So it's very striking. And just to make this point in a different way, this is just looking at all pairwise combinations of all those alleles. You find that just in that first exon, that first domain, there's about an average of 54 differences between alleles in all pairwise combinations and 33 amino acid differences. Okay, so there's only 100 amino acids there, and 33 of them tend to vary on average between alleles. Okay? They're very different from each other. And this is just to make the point that most of those alleles are actually present as single copy or duplicates or triplicates in the population. So lots and lots and lots of alleles, very low copy number for each one. Okay, and so if you put those two things together, the rules I just told you about and this high level of polymorphism, it's not surprising that if you go out and just take colonies at random, 98 to 100% of the time they're going to reject. Only 0 to 2% of the time do you see fusion or even transitory fusion responses. Okay, so it's a very precise system. So that's what we've known, and now I'm going to sort of transition into talking about work we're doing to try to figure out how do these proteins actually do this, right? So it's enough, we have rules, we can predict the outcome based on sequences, but how these proteins actually make that distinction between self and non-self. And so we came up with a really simple hypothesis, and that was if these things are sitting on the cell surface, and they're the proteins that are involved in it, perhaps involved in allorecognition, perhaps the way they work is that if colonies have matching alleles, those proteins will bind to each other, and that somehow tells the colonies to fuse. And that if the proteins don't bind to each other, that leads to a rejection response. Okay? So it's a simple hypothesis. And there's a way you can test this hypothesis. We couldn't, unfortunately, do it in hydroactinia because it's hard to work with hydroactinia, but we could take these proteins out of hydroactinia and express them in cell culture. Okay? So now we're moving from hydroactinia to mammalian cells. And fortunately, mammalian cells express these proteins fairly easily, but they express them. Okay? So if you transfect mammalian cells with a protein, with one of the ALR proteins, and there's a tag on the end so we can be sure that it actually gets expressed properly and it's sitting on the cell surface. And then we co-transfect in a GFP, which just lets us tell by eye which cells are expressing those proteins. We have a dish of cells that are expressing the proteins. We grow them up for 48 hours, and then we actually dissociate those cells into a single cell suspension. And then we keep them on a shaker. And if nothing else is going on, those cells will remain as single cells in suspension. And if you look at them under the fluorescence microscope, you'll see that some of them are expressing the marker. So if this would just be if the cells were just expressing GFP. It's a transient transfection, which means not every cell is expressing your protein. And so we don't see every cell green, but we see most of them. But if these ALR proteins are capable of binding across cell membranes between proteins, the cells will actually start to stick together. And you would see, you would see clumps like this. And I just sort of showed you the results. And so, and so that's what we saw. So this is an example of our controls. This is just taking two cell lines, one expressing GFP and one expressing cherry with nothing else, M-cherry with nothing else. You put them in suspension, you get these single cells. They stay as single cells. If the cells are expressing either the F allele or the R allele of ALR1 or of ALR1, they'll form these nice big clumps. Okay? Now the real question was, that's great, so these proteins can bind to themselves, but did they do it in a specific manner? So in other words, does ALR1 always bind to itself, and ALR1, or the F allele always bind to itself, and the R allele always bind to itself? And when we did those experiments, we found that they did indeed. So this is a mixture of a cell line expressing the F allele in green and the R allele in red, and you can see they form these nice big clumps. They're segregated by color, and this is just to show you that if you keep the alleles the same but vary the color, you do get these multicolored aggregates. We are able to detect homophilic binding that way. Okay, so we've tested a handful of alleles now in this system. 6 alleles for ALR1 and 4 alleles for ALR2. We actually have more, but this is what's been published. And in all cases, the alleles are able to bind to themselves. That's this blue line on the diagonal here. But they do not bind to other isoforms. With one exception here, this is an example where there are 2 alleles, and they're only differing by 3 amino acids. And most of those amino acids are very close to the transmembrane domain. Okay, so you don't have to have a perfect match, probably because not every part of the protein is involved in that interaction. And so we think that this is what's going on in vivo. Okay? And we think it's probably a little more complicated than just this, but we think these are the proteins that are involved in making this decision between fusion and not fusion, or rejection. So one thing you might be thinking is, okay, so you just told me that there's 183 different variants out in nature. Is it possible for a system to evolve 183 different binding specificities for a protein? Or how do you evolve 183 different alleles where every allele will bind only to itself, but not to all the other alleles that are out there? This is more concisely than I just said what I mean. And so this has actually become a sort of a central question in my lab, is trying to figure out what mechanisms are at play here to generate this diversity. And we think there's probably three things going on. And I'm going to show you some preliminary data for each of these. We're actively working on this, so this will be where we're at for each one of these. But one thing you might imagine goes on is recombination or gene conversion, and I'll explain what that is. Mutations, just plain old accumulation of mutations over time, and then also variation in the number of genes that are actually present in between different animals. And I'll talk a bit about that also. So first, recombination and gene conversion. So you might imagine that if you look at the locus, actually one thing that's sort of striking about this locus, this is now looking in the genome. So not at the protein, but how the gene is encoded. So each one of those IGSF domains is encoded as a separate exon in the genome, which is kind of interesting because it means that you might very easily be able to have recombination occur between two alleles, say for the blue allele and the green allele here, and you can mix and match these IGSF domains so that you ultimately end up, if recombination occurs, with a new allele just by swapping these around. And the question is, would this new allele have a new binding specificity? Okay. And so we went out in nature back into that, we went back into that pool of alleles that have been sequenced. And we looked for cases where there were alleles where everything else was the same except for this N-terminal domain here. And we found 3 examples, 1, 2, 3. And then we tested them in vitro, and sure enough, each one of those only binds to itself and not to the other allele. So just varying in this first domain here is sufficient to prevent alleles from binding to each other. And so recombining those domains would be sufficient to create new binding specificities. Now, we haven't done this extensively, but I have a grad student who's right now working on not just looking at what exists in nature, but taking different alleles and then systematically swapping these domains around in vitro to see whether we can sort of create new binding specificities and narrow down which domains in this ectodomain are important for the binding specificity and which ones might not have any role in that. But regardless of what she finds, just looking at that first domain, we think that this actually happens quite a bit in nature. And the evidence for that comes from, again, some work that was done looking at the genomic locus and then the population. So if we this is the gene I just showed you. Okay? If you look in the genome upstream, there are 2 pseudogenes, which are pseudogene 1 and pseudogene 2. And what these are actually partial duplications of this part of ALR2. Okay? So in each hydroctinia on each chromosome, there's actually 3 copies of this first domain. There's the functional copy, and then there's 2 pseudogene copies of that first domain. And what Andrea did when she did that big population study was she actually sequenced not only the expressed allele, but also the pseudogenes from the genome. And if you make a phylogenetic tree of those sequences, what you find is that very often the domain 1 sequences that are in expressed genes, so these are things we know are actually genes, are most similar to pseudogenes rather than other expressed genes. Okay? This is exactly the pattern you'd expect to see if in nature these sequences are getting donated or they're getting swapped around a lot by recombination or gene conversion. Now, at this point, we don't know what the mechanism might be for this, but it's really, it's kind of an intriguing finding. Oh, and sorry, this is the graphic that goes with the fact that my grad student's working on figuring this out. So in terms of mutation, we've started to look at this a little bit too. And so what we've done is, since we know that this first domain in and of itself is sufficient to affect whether the proteins will bind to each other, we've gone and just taken all of those domain 1 sequences, and we've looked for instances where there are very closely related domains that we know don't bind to each other. Okay, so I'll show you one example of this is Just ignore the names here. They're not really that important. But we identified from the pool of alleles that we have in the lab 2 alleles that only differ by 6 amino acids, and they do not bind to each other. Okay? Now, there are other alleles in nature that are 1 or 2 or 3 amino acids different from these. And so the thinking is that somewhere during evolution, whatever the ancestral allele is here evolved to form this allele, but also evolved to form this allele. And at some point, the binding specificity changed. Okay? So could we narrow down where in evolution that binding specificity changed? And so one thing we can do is use an approach called ancestral sequence reconstruction to actually go back in time and sort of go back in time and figure out what was the sequence of the ancestor of these alleles. And if we can do that, we can make an alignment of not only the existing sequences, but these ancestral sequences, identify where those mutations occurred, and order them in time over this tree. And then we can synthesize these sequences in the lab and then test them and see what their binding specificities were. And in that way, we can figure out exactly where along here and what mutations were key for creating new binding specificities. This is important because we want to understand whether single mutations could result in new binding specificities. So you could have not just recombination, but also mutation leading to the generation of diversity in the system. And this is kind of a little bit of a tangent, but in doing this, we started to wonder, well, so if you look at, if this is just the linear sequence of the amino acids, but of course these things are folded in three-dimensional space. So if we were to map these differences onto a predicted structure, would it tell us something about how these proteins might be interacting? And so we did this for a couple of different single alleles. So this is just the first domain. This is the surface representation of the predicted 3D structure of the protein. And then each blue spot is where one of those variable positions is. And what we found is that in both of these cases, it looks like all the variations on one side of the protein, not on the other, right? And so we went to that big sample of alleles, and we then mapped all the variation in, well, we had 139 complete allele sequences. And when you map all the variation on that, what you see is that one side of this domain has nearly all the variation. The back of it's completely conserved. And if you look at the second domain down on this protein, what you find is that there are these little hotspots of variation. The back is fairly well conserved. And by the time you get to the third domain, there's really no clear, obvious hotspot for the variation. And so what we think this means is that all the interactions are going on here and potentially here. And if you think about sort of how these proteins might actually be interacting when they're coming at each other from different cell surfaces, there's lots of different possibilities. We think this one might be the most likely, where you have the proteins sort of bending, coming together. So that first domain, that first domain has a homophilic interaction. There's a little bit of an interaction between the second domain, and there's nothing going on between the third. This is, of course, speculation. And like I said, we're working to actualize these things and to be able to test this empirically. Okay, so the final thing that I want to tell you about in terms of generating diversity in the system has to do with gene copy number variation. And this is based on some data we just got in the last couple of weeks, which came out of a Genome Project. So my lab is collaborating with a couple of other labs that work on hydroactinia Institute, actually at the NIH, and we've sequenced hydroactinia. And as part of that, we've actually gotten the sequence of not just ALR1 and ALR2, but the whole region surrounding ALR1 and ALR2. So going back in time, when we first identified ALR1 and ALR2, this is our genetic map. We had done something called chromosome walking with bat contigs. They're basically like DNA plasmids with very large inserts, 100 kb or so. And we had sequenced regions around ALR1 and ALR2, but we didn't have the full sequence. Now with the Genome Project, we've gone from having about 3 million base pairs of sequence to 11 million base pairs of sequence, including essentially the whole thing. There's 2 little gaps here. I'm not too worried about those. There's a region where there's a lot of repeats, and I think that's just hard to sequence through. So this has kind of been like Christmas for us, because it's like, oh, there's all this data, we get to go and mine it. And so the first thing we did was we looked for ALR1 and ALR2. So ALR2 turns out to be exactly what I just told you about. There's nothing new. But ALR1 has been full of surprises. So if you take the sequence of ALR1 and you blast it against this region and look for where it is, it's right here. And then we thought, well, maybe there's other ALR1s. And so just to be sure, we took each of these domains separately and looked for sequences that were similar to each of those domains. And what we found is that surrounding ALR1, there are 10 regions that look like they encode full ectodomains, so the IGSF1, IGSF2, and the spacer, as well as partial sequences of these other domains. Now, at this point, we don't know if these are actually genes or pseudogenes. But if they were, you might imagine that it's not just ALR1 and ALR2 out in nature that are controlling allorecognition, but there's other genes where there's variation at those, and those also are contributing to the response. Now, the real surprise was that further upstream, we found another ALR1-like gene. And this one's only about 30% identical. And surrounding it are, again, a cluster of 11 ectodomains and 6 partial domains. And again, we don't know whether these are actually expressed genes or not. But what this is starting to look like is a system where you can have variation not only at the level of the sequences of these genes, but also in terms of the number of actual genes you have that are controlling the response. So in other systems where you see things like this, so for example, there are these cells in the immune system called natural killer cells, and they function by a similar recognition system. If you sequence 2 different individuals, they actually have different numbers of those genes that control the self-recognition system. And we think that this is probably going to be what we find here. The other thing that is striking about these, or that has been interesting about these, is we wondered, could there be a lot of shuffling between the sequences of this ALR1 gene and all these ectodomains? And so a simple way to do that is to basically take each of these parts of the domain gene, and then we've only sequenced one animal, so we only have sort of this reference sequence to look at. But what we do have is the sequences of ALR1 genes from other colonies from out in nature. Okay? So the question is, are the sequences from nature more similar to ALR1, or are they similar to these other genes? And so this is just showing you the names of all those alleles from nature of ALR1. And we call them ALR1 genes because they all have this conserved cytoplasmic tail in the transmembrane domain. So if you take the sequence of each of these and you do a similarity search against this reference genome that we have now, what you find is that the top hit is always the bona fide ALR1 gene. Okay? It's not the case if you look at this first domain. So if you take the first IGSF domain and you ask, okay, which in these wild-type alleles, which sequence is most similar to that N-terminal domain, it turns out a lot of the time it's these other non-ALR1-like genes. Again, this is exactly the pattern you'd expect if there was lots of recombination going on between these genes. The same is true for the second IGSF domain and the spacer. Okay? And so what we think is what we think is happening at ALR1 also is a lot of recombinations generate diverse new alleles. So I'm going to wrap up in like 3 or 4 slides, but this suddenly got much more complicated than we remember. When I started, I would say there's 2 genes. Now there's potentially a lot of genes, and we're working in hydroactinia, so it's kind of hard to do genetic manipulations. Recently, this has changed because of something called CRISPR. So some of you may have heard of what CRISPR is, others maybe haven't, but so I'll give you, this is the only slide explaining it. But basically what it is it's like a restriction enzyme, but the cool thing about this is you can program it by giving it a short sequence of RNA that matches a target in your genome to go and cut wherever you want in the genome. Okay? And when it does this, it can induce mutations, and you can do, and therefore you can make targeted deletions of genes in the genome. And it seems to work in pretty much every organism that it's been tried in, at least in, with any effort. And so, the people who work in mice and things were really excited about this because it meant they could do some things a little easier. For folks like us, this was huge because we went from not having any genomic tools to now having genetic tools. And so if you went to like conferences and went to the bar afterwards, the conferences I go to, you would hear conversations like this where somebody was saying like, I got CRISPR working in my organism, and me too, right? So this is our Me Too slide. So we've been working with Cas9, and the way we do this is we take those embryos and we have a microinjection system, so we inject them with the Cas9 protein and the guide RNA. And we started out by targeting ALR2, mainly because it's the one we know the most about, and there's only one of them, and so that made things a little bit easier. We didn't target this ectodomain because, as I told you before, it's been duplicated, so it was tough to find some targets. So instead, we focused on trying to knock out the transmembrane domain. And the idea here is that if we could cut out this region or disrupt this region, the transmembrane domain would be disrupted, and even if the protein was expressed, it would be secreted and hopefully be functionally like a knockout. Okay, so we could then test its function. And so the way we did this was if we injected Cas9 that would cut two different pieces in two different places around that domain, it would essentially act like a pair of scissors and cut out this region and thereby disrupt the gene. And so we injected those single cell embryos. We then pooled the larvae just to see whether it's working or not. And then by PCR of the surrounding region, we looked for colonies that had, or larvae that had a shorter piece of genomic DNA than you would expect in the wild type. And we saw evidence that this was happening here, here, and here. And we cloned a subset of those and found that indeed, if you look at, here's the wild type genomic locus, and then when you sequence this region, you find that exactly where you expect to see the cut site, there's a deletion here of about 2.2 kb. So it works in hydroactinia. Now the problem is, in order to get all this data, we had to grind up the larvae, and so we didn't have them anymore. And so we've had to go back and do more injections. And so my postdoc, Steve, has been working on, he's injected a whole bunch of colonies. We now have about 11 of these that are growing in the lab. And what we want to do is just so these colonies, because we don't know where that mutation actually happens during the development of the organism, they're going to be mosaics. So they're going to have some functional loci and some non-functional loci. And so what we want to do is then breed these guys and hope that some of the germ cells actually carry those mutations, and such that the offspring will then be completely mutant for whichever mutations we happen to have induced. And then we'll test these. And the idea is to test whether we can functionally change the phenotype by knocking out one of the genes, and does that conform to our expectations. So this is work that's ongoing. So stay tuned. And so just to sort of sum up here, I hope that I've convinced you that allorecognition is controlled by these two genes. And that the way they seem to work, at least in vitro, and we think in vivo, is that they are homophilic binding proteins that have a really remarkable specificity, and that there's hundreds of alleles out in nature, and that by recombination, mutation, and perhaps copy number variation, there's a huge diversity of allorecognition specificities, and that's what allows the system to be so specific and let these colonies do something that's pretty remarkable, right? They can tell self versus non-self just by touch. And I want to make two final points. One is that if you look around in nature, there are lots of systems like this. Okay, so